Hello students, here's a quick presentation on the regulation of the Trp Operon. So like many bacterial genes, Trp Operon is a collection of different coding regions for a variety of different enzymes involved in tryptophan synthesis. And since the cell wants to have all of these available, at the same time, it makes sense to put them under the same regulatory sequences. Now, in the case of the Trp Operon, there are two important regulatory methods that are involved. First is based on the operator of the gene. So you see the operator region is able to bind a protein called the Trp repressor. Trp repressor is produced by the TrpR gene, which is produced somewhere else in the genome. It's acting as a transacting factor. And so when there is a lot of tryptophan around, the tryptophan, here represented by its by the symbol W, right, is able to bind to the Trp repressor, and together they're able to bind to the operator. And when it's bound to the operator, the RNA polymerase still comes to the promoter, right, and it tries to go through, but it gets the operators in the way. So as a result, there is no gene expression. None of these TrpE, D, C, B, or A get expressed. On the other hand, when you have a low amount of tryptophan, right, there is not enough tryptophan around to bind with the repressor protein. And as such, the repressor protein is unable to bind to the operator, so the RNA polymerase is unhindered and is able to travel through the gene. The other method of regulation for the Trp Operon is using the attenuator. So this takes advantage of the fact that that in prokaryotes, translation occurs at the same time as transcription, right? So the RNA polymerase moves through part of the gene, right, and produces its mRNA transcript. Now this mRNA transcript is still attached to the polymerase, and the polymerase is going to continue to polymerize that, unless the attenuator gets in the way. Now, so it produces the mRNA, and the ribosome immediately comes on to attach. Now, if there's a lot of tryptophan in the cell, this ribosome is going to start to read it, and it's going to come across two tryptophan codons right in a row. Now, if it's able to put those tryptophan in right away, just because there's a large concentration of tryptophan in the cell, the ribosome, the attenuator sequence in the mRNA, is going to change conformations, and it's going to change into this terminator hairpin. And the effect of this conformational change in the mRNA is that the RNA polymerase terminates transcription. It leaves the gene, and this is all you get. And this is non-functional mRNA. It doesn't contain any of the structural genes, just this leader sequence. On the other hand, if tryptophan is low in the cell, the ribosome will get to those tryptophan codons, and it will stall. It will be unable to fill in the it'll be unable to fill in the it'll be unable to elongate the chain. As a result, the RNA will change conformations into a different conformation, which is called the anti-terminator hairpin. Now this loop doesn't affect the RNA polymerase, so it's able to travel through the rest of the gene and produce an mRNA that contains all the other structural genes in it. Thank you for watching my presentation on the Trp Operon. Have a wonderful Thanksgiving, and remember, the turkey does not actually contain more tryptophan than any other type of meat, so tryptophan is not the reason you feel sleepy after eating Thanksgiving dinner. It's just because you ate too much.